Go. Hey everybody, Dr. John here for Extract Lab. How are you doing today? Today we're going to be talking about some of the requirements for GMP extracts. There's a lot of confusion because there's a tremendous recreational market out there. A lot of our customers are trying to create API or active pharmaceutical ingredients or botanical intermediates or herbal medicinal intermediates. And a lot of the definitions and things like that are in the recreational market are kind of making their way into the pharmaceutical world. And it's kind of interesting because it creates some confusion because sometimes there's no definitions for things that are really rooted in science. So we're going to unpack that a little bit, and we're going to be looking specifically at full spectrum extracts and what that really means, what the requirements are for GMP extracts. So let's get started. So the first thing is, what are the key requirements for creating an active pharmaceutical ingredient or a botanical drug ingredient? And what are the key things that you really need to be thinking about? Well, I came up with a list of 4. Number 1 would be stability, the stability of the extract itself, to maintain its profile, both purity, potency, identity, over time and in storage conditions. And even when it's taken into the body, there are some stability thoughts there that need to be really understood. The second one was consistency. How consistent is the potency? Obviously, you can't have a dosage form that changes from dosage form to dosage form. So how consistent is it? Third thing is a consistent impurity profile. What that is if you have an extract, for example, if it's coming from a botanical, you might have a very low-level impurity profile, and that needs to be somewhat consistent over time as the extractions go on. And that's why a lot of people do purifications. That's why a lot of people remove the more reactive components of an extract. And so a consistent impurity profile, and then also an unambiguous therapeutic effect. So, for example, if you have one particular active pharmaceutical ingredient and then you have another active pharmaceutical ingredient, those are called combination products, you want to be able to understand what those two components do to, give an effect over time. And if you have thousands of compounds, some of those compounds might, for example, have a therapeutic effect, but you wouldn't even know it. Or it could be just the opposite. It may have an adverse effect, but you wouldn't really know it. Those are the kind of the key conditions for GMP extracts. Is it stable? Does it have a consistent profile to it? Does it have a consistent impurity profile? And is it an unambiguous therapeutic effect? So those are the key things that you really need to be thinking about when you're talking about GMP extracts. Okay, so how does this impact full spectrum extracts? Well, it really impacts it from how you're making the full spectrum extract. There is no compendial definition, meaning official scientific definition of a full spectrum extract. You need to know that. There are no, like if you go into the European Pharmacopoeia, you will not find a definition for a full spectrum extract. That's because it's not a scientific definition. It's a thought. It is a more or less a recreational thought. What the European Pharmaceutical or European Pharmacopoeia does have are definitions for standardized and quantified extracts. And there are several medicines that are out on the market right now, both in the US and in Europe, that would qualify under both standardized and quantified extracts. And those would be Epidiolex or Sativex. There's this thought out there, a lot of people consider Sativex to be like a full spectrum extract, but it's not. It's actually formulated. It's a formulated extract, and actually the formulation is not known, but we do know it's formulated. So it's not necessarily a full spectrum extract. Now, the best definition that you can get, the closest you can get, is from a industry group called the AHPA, or the American Herbal Association. And what they do is they serve the nutraceutical market, and they came out with a compendial definition of what full spectrum extract is, and that is, number 1, it's an extract where there's not a lot of processing associated with it. There's not any major components removed from the extract itself, and it usually has the full spectrum of everything that's in the plant. So, for example, if you have a broad spectrum, if you have a lot of different terpenes, it would have all those different terpenes in it. If you had fats in it, it would have the fats in it. If you had the waxes in it, it would have the waxes in it, according to this definition. If you had a broad set of cannabinoids in it or flavonoids in it, it would also have those. Now, given the nature of that definition with the GMP requirements, they're kind of at loggerheads, if you know what I mean, because you want to have a stable formulation or you want to have a stable extract for a GMP, but the AHPA is saying, hey, if you want to have a full spectrum extract, you're not going to be processing all of those things out of it so that it will be stable. For example, you're not going to process out, or you're not going to remove the waxes, or you're not going to remove the fats, or you're not going to decarboxylate or things like that, which most all companies who are in the pharmaceutical world are going to do any combination of those things. So you can see that the idea of a full spectrum extract and its roots not only in the dietary supplement world, but also in the recreational world, don't really fit very well with the concept of GMP extract. I'm going to kind of unpack that a little bit more. Let's explore, for example, together the idea of a consistent impurity profile. Okay, so I take an extract, I pull it out of the plant, and I start to measure it, and I got the major components of the plant. Let's just say it's THC and CBD. Okay, so I got a ratio of THC and CBD. In the HPLC chromatogram, it looks pretty clean, but then as soon as I move it over to the GC, I might find a whole terpene, a whole bunch of different terpenes in there. I might find flavonoids in there. I might find a lot of different components in there that I wouldn't have seen in my HPLC, but I know that they're there. That kind of fingerprint is what's known as the impurity profile. One of the main issues associated with full spectrum extracts is that it has these other components in there. They're not very stable. And one of the key aspects of any pharmaceutical is that it's stable under conditions of use, number one. And then number two, it's also stable under storage conditions. When you have full spectrum rich terpene profile, that is highly, highly reactive. It's always changing. And that fingerprint, if you will, is changing over time as it's being stored. Even if it's stored under very stringent conditions, no light, for example, or stored at a lower temperature, just the very nature of the terpenes, they have these double bonds on them, and they tend to react and degrade into other terpenes, or they may combine with each other. So there's a lot of different things that happen there. Really, the issue with the reactive molecules in an extract, you want to try to get those reactive molecules out, and you want to try to give yourself a consistent purity profile. Now, on the other hand, you can engineer with natural components a terpene profile using stable terpenes that have been purified. And those actually will produce pseudo full spectrum type of GMP extract, but you won't have all of the issues associated with terpenes that are not stable. The other thing you want to think about is, there's this whole idea of the entourage effect, and everybody talks about it, especially in recreational markets. Well, the entourage effect, what that is that's the synergistic action of multiple components within a whole plant extract that would give a good benefit. Like, for example, okay, I have 7 different terpenes and CBD and THC, and we're going to fight inflammation. So that's the idea, and it's all working together, but we don't really know why, okay? Or we haven't really figured it out because there's thousands of different components in that whole extract, and you wouldn't really know. Maybe you would have some that were working really well and others that were not, okay? Some of them were fighting inflammation. On the other hand, some might be causing inflammation. You don't know. So the purpose of bringing a botanical drug into the marketplace is to establish the efficacy and also the effects of all the different components on the condition that you're trying to address. If it's not really ambiguous, if it's not unambiguous, meaning, okay, well, this component is affecting it, and this is my API, and that's also affecting it, well, which one is doing more, right? Or maybe they're both together. Maybe they're both doing it together. But on the other hand, maybe this one's degrading, producing a product that then is counteracting it and causing inflammation. What is required in any drug development process, and botanical drug development process or herbal medicinal drug development process, would be to really unambiguously define what the effects of formulation is on the actual indication that you're looking for. That's what I mean by unambiguous therapeutic effect. And it's a really important factor. You don't want to get bogged down in your formulation and find out that, okay, well, we have changing impurity profile. We got to know, okay, well, now we have to find out what all those unknowns are in there. Some peaks are showing up that weren't there before because it's not stable, because we're using full spectrum extracts and it's not very consistent and it's not stable. And okay, now what are we going to do? I would encourage those who are in the GMP world, who are looking for GMP extracts, to go into a little bit more refinement. There's the whole idea, well, they're going to create a bunch of full spectrum extracts. We're going to sell it as a Sativex-like surrogate and we're going to make a lot of money. Well, no, not really. You are assuming that Sativex is a full spectrum extract, where in fact, actually, it's not that, because it's been formulated. It's probably a quantified extract and a standardized extract. And in order to get those types of extracts, you're going to need to make them stable. You're probably going to have to distill. You're probably going to have to do some separations. And you maybe even take a look at maybe formulating in some of the stable terpenes rather than having a broad spectrum of unstable terpenes. So that's the bottom line, guys. I hope you guys enjoyed this. The bottom line is really that full spectrum extracts are not GMP, and they can be made into GMP products by further processing downstream and making them really fit the criteria for a GMP extract. So this has been Dr. John for Extract Talks. I hope you guys had a good time, and thanks for joining us. Take care.